Good morning or good afternoon, everyone. I'm Sabrina Buqua, Chief Marketing Officer at Inova Software. First of all, I would like to welcome you to our first therapeutic-specific virtual partnering event on oncology, bringing together top oncology companies from across the globe for 3 days of online face-to-face meetings. This event is part one of the virtual partnering series BIO and Inova have decided to launch. We believe that there is a great need for more high quality and targeted partnering events and a need to unite specialized business development and scientific communities with specific therapeutic areas to bring the best science forward. But before we get started, just a few administrative logistics. So everyone's on mute to minimize background noise during the webinar. Please type your questions in the questions panel on GoToWebinar. And lastly, this webinar is recorded, so we'll send you a link to the recording. And it is now my honor to introduce the moderator of today's plenary discussion, Dr. Stephanie Oestreich, Venture Partner at RA Capital Management. Stephanie's primary responsibility at RA Capital is to identify compelling assets for new company creation. She has extensive biotech experience. Most recently, she served as Executive Vice President, Head Academic Partnerships and Investments, North America at Evotech. She also has experience at Hoffmann La Roche and Novartis in various leadership roles. Please join me in welcoming our esteemed moderator, Dr. Oestreich.
Thank you very much, Sabrina. Good morning, good afternoon to everybody. We're very excited to have a really esteemed panel today with us, and we will cover in this morning's discussion some of the latest developments in oncology R&D, including some real-world evidence, also some advancements in cell therapy, and some of what was hot at ESMO, and also some perspectives on the investment landscape in oncology, obviously without giving any investment recommendations. And lastly, some points on AI and machine learning. We have with us today a really great panel from Europe and also the United States. Elisa Kiebach is CEO and co-founder of the TCR company Tknife, and she's joining us from Berlin, Germany. Dana Fuchsbaum is Director of Product Management of Clinical Genomics at Flatiron Health. And Zachary Hornby is CEO, President, and Director of Boundless Bio, joining us from San Francisco. Elisa, would you like to start to introduce yourself, and then Dana, and then Zach?
Of course. Thanks, Stephanie. Good morning. I'm Elisa Kiebach. I'm the CEO and co-founder of Tknife. My background is in immunology, cell biology. Before joining Tknife, I was an academic researcher working with T-cell receptors and T-cells and how we can apply them in the treatment of tumors and autoimmune diseases. And in 2018, we founded Tknife, a Berlin-based TCR and cell therapy company. We have, as our core technology, specialized mouse model for the generation of therapeutic T-cell receptors. We are preclinical stage and developing cell therapy in the context of tumor.
Great. Thank you, Elisa.
Thank you.
Hey, everyone. Good morning. My name is Dana Fuchsbaum. I'm a product director at Flatiron Health, which is a real data company based in New York City. Very much looking forward to the dialogue today, and apologies we can't see folks over video. So just by way of a very brief introduction, I've been at Flatiron for just over 5 years. Flatiron is a two-sided business. On one side, we build software for medical oncologists, the primary piece of software being our electronic medical record. And then on the research side of the business where I sit, we curate real-world research data sets from that underlying electronic medical record data. So the product that I currently lead is our clinical genomic database, which is a product that links real-world EMR data, so things like demographics and outcomes and treatments, with comprehensive genomic profiling data from Foundation Medicine. So that's a product we've been building out for the last 2 years, and it's sort of the cornerstone of our precision medicine, our precision oncology strategy.
And good morning. Hello, I'm Zach Hornby. I'm the Chief Executive Officer at Boundless Bio. My background is that I have been working in the biotechnology industry for the past 20 years, the first 5 years in Boston, and the last 15 years in San Diego in various operational roles. And for the past 12 or so years, I've been working in oncology companies. Most recently, I was at a company called Tknife that I helped form, initially served as the Chief Financial Officer there, and then the Chief Operating Officer, where we were overseeing a pipeline of multiple development stage targeted therapies. And we eventually brought one to market globally, a TRK and ROS inhibitor called Rozlytrek, which led to our acquisition by Roche. And one of the learnings we had with that experience was the power of precision medicine within oncology, but also its shortcomings. And so we understood that precision medicine and targeted therapies work really well for certain types of oncogene alterations, like gene fusions, activating point mutations, and certain skipping deletions. But we saw that the one place where precision medicines was really falling short, it was with the patients with gene amplification-driven tumors. And so we started this new company, Boundless Bio, explicitly to try to address that major unmet need within the oncology field, gene amplifications. And part of this science by which we're doing that is through our realization that gene amplifications frequently occur in a different location within the nucleus of the cell. They actually do not occur on chromosomal DNA, but rather reside on a separate piece of DNA called extrachromosomal DNA, or ecDNA. And it's through this understanding of ecDNA biology that we're beginning to better understand how cancer cells perform and propagate, and how we might identify vulnerabilities in them that we could therapeutically exploit. And so that's what we're doing at Boundless. Pleased to be part of this panel.
That's great. Thanks to the panelists. And let's jump into some questions. Dana, let's start with real-world evidence. So it's being used right now to glean information about patients with molecular subsets of cancers. Can you speak a little bit about the challenges and limitations of collecting real-world data, and also talk a little bit about the fact that many clinicians are, all of them are accepting basically phase 3 data as the gold standard. So how are real-world data being used in contrast to phase 3 clinical data?
Sure, yeah, very much happy to talk about the challenges and limitations. So, I think the first one that comes to mind is in collecting real-world data, we are limited to the testing modalities and the testing patterns that are used in routine care. So for instance, at Flatiron, we see a lot of community oncologists in our network. And so, right, the variability of testing is quite large. And so in an ideal world, you would be able to have a research data set where you have a baseline sample and then subsequent follow-up, as well as serial, blood draws, pre and post treatment. And that frankly just doesn't happen in the real world. And so you have to be mindful of these testing patterns when evaluating this type of data. I think one of the things that also is important to remember is methods, right? Methods are really, really essential to analyze this data properly. Real world data is a cross-sectional convenience sample. We tend to see longer surviving patients represented, which results in a survivorship bias, right? Patients must survive long enough in order to get testing. And so that's another piece that when analyzing this data, you have to think a lot about and use the proper methods to account for that. I think another piece, and this isn't necessarily just related to genomic testing, but thinking about the utility of real-world evidence in the context of gold standard clinical trial data, right? We must work to get to as much of an apples to apples comparison. And I think where we find challenges is curating meaningful endpoints. So, right, in the clinical trial setting where you are looking primarily at RECIST, we find that in routine clinical care, RECIST just isn't captured, right? And so we need to develop approaches to curate endpoints that are still meaningful and importantly valid. They might not be RECIST, but do they confer, some increased understanding of a patient's response to treatment is a big challenge at hand in leveraging real-world evidence. And then the final piece I'll say, I can go on for a long time, but I'll keep it short, is, one of the use cases that's emerging for this type of data is demonstrating unmet need, right? These are really rare populations. Sometimes in trials, we're seeing, double digits in terms of the patients with a specific genomic alteration. So real-world data allows us to expand the purview a little bit and ideally find more patients. But when we are doing these comparisons, it's important to recognize what are we actually comparing, and to the earlier point, can we replicate clinical trial criteria in the real world so that we're doing an accurate comparison and really kind of demonstrating what is needed to support the evidence package for that specific drug. I'll pause there. I'm sure there will be follow-ups.
Thank you, Dana. These are really important innovations here in this context, and you and maybe also Zach can comment on that. On the one hand, we are able to be more and more precise for even single patients about gleaning data about their condition. At the same time, we're trying to develop therapies for as many patients as possible. Can you talk a little bit about these two, seemingly conflicting or contrasting goals?
Sure. Sounds like that question was directed to me, Stephanie.
Yes.
Yeah, that was something that we initially struggled with a bit at Ignita and are now thinking about a lot at Boundless. And the way you try to reconcile those 2 points is the precision versus the small numbers is ultimately what's the common factor that's druggable. So for instance, if you take a given alteration, let's say an oncogene that most people are familiar with, EGFR. Well, it turns out there's lots of different ways that EGFR can be altered. There can be a common point mutation, L858. There can be amplifications. There can be certain deletions, like the exon 19 skipping deletion. So not every mutation of this gene is the same. And if you start getting more precise about the nature of the mutation, then you start slicing the salami more finely, and so you're getting smaller and smaller patient populations. But it does turn out that for this particular gene, EGFR, that most of those alterations are all activating in such a way that a kinase inhibitor is going to work irrespective of the specific activation. So you can lump together some of the point mutations and deletions and call these EGFR activated patients and treat them with an EGFR inhibitor like the drugs Tagrisso by AZ. However, there are other types of EGFR amplifications like gene amplifications that don't respond to the EGFR inhibitors. And that's because the biology actually behaves differently. So it's not about the structure of the protein or the shape of the molecule. It comes down to how the cells actually employ the gene and the protein product. And in the case of the gene amplifications, which, as I talked about earlier, reside on extrachromosomal DNA, the cells have this unique and sinister way of they can upregulate or downregulate various different genes on the ecDNA. So if you apply therapeutic pressure like an EGFR inhibitor, the cells actually have the ability to eliminate their reliance on EGFR and instead decide to become reliant on a different oncogene. And so you can see that in that way, any individual therapy may not be effective if the cell keeps switching its dependency. So this is a bit of an example that helps highlight the question of how do you know when through genomic capabilities like Flatiron and others have, how do you know that you've actually found the specific characteristic of the patient's tumor that you want to put it into a certain bucket or category for treatment? And I think that's what the field is really honing in on. And it's a complementary relationship between the diagnostic providers like Foundation Medicine, the data aggregators and interpreters like Flatiron, and then the drug discovery companies like Boundless Bio, who are understanding the biological significance of any given tumor characteristics. I'll stop there.
Mm-hmm. Thanks, Doug. These are all some great advances. Elisa, another very interesting field is cell therapy. Can you talk a little bit about the challenges you're facing as a young cell therapy to start with, and then also tell us a little bit about the recent developments in the cell therapy field and how that is impacting your company's strategy?
Sure, absolutely. Yeah, I think so. When we speak about precision medicine, it seems like TCR therapy, what we do, it's already a very precise drug that we're developing, right? It's a T-cell receptor targeting a very specific 8 to 10 amino acid epitope, which is bound to very specific HLA. So we're targeting a very specific subgroup of patients with a very specific target. And the question as a company for us is still there are many different strategies that how we can tackle this type of therapy and develop a product in the end. And of course, this is since we're so from start focused on the target, we have to ask ourselves which is the right indication to go after with this target. So which indication would, maybe be most susceptible for the type of cell therapy that we're doing. So is it a high chance for clinical response, or is it rather that we should go after an indication where there's a larger market or the highest need? So of course, these are all strategic positionings of the company that from beginning on is really the main thought process that we have to go through. Yeah, and I mean, we talk about oncology, but the platform company that we are, we also ask ourselves frequently, is there, for us as a company, value and maybe going beyond oncology, moving into field of infectious diseases, autoimmune diseases, where the same type of cell therapy approach could, of course, also be tested and has been tested by other companies as well. Or moving beyond the cell therapy format. So T-cell receptors, as you know, have been developed in very other formats as well. So why not use it as a soluble molecule, as a TCR engager, for example, similar to the BiTEs, which are currently developed. So and this of course seems far more attractive to people who are more used to work in the biologics field and as opposed to cell therapy. So many different routes with the company where we could go to, and even if you stick to just looking at cell therapy, maybe we have to decide ourselves, do we want to develop a very patient-specific product? Is it an autologous product which is only developed for this one patient, or are we testing allogeneic approaches where we have an off-the-shelf product that can be administered at least to a certain number of patients? So as a very young company, this strategic positioning in the entire space of cell therapy or T-cell therapy in oncology, these are really critical thoughts we have to go through and really make sure we don't lose the focus as a company, but also, of course, remain open to new developments and not to miss out on certain opportunities, I would say.
Mm-hmm. Thanks, Elisa. It really seems that young and more established companies are grappling with a lot of similar issues. One that is probably very pertinent for cell therapy is manufacturing. Can you tell us a little bit about the challenges there and how you're tackling those?
Sure. So I mean, manufacturing, the process is always considered at least 50% of the final product that we're doing in cell therapy. So it's really critical to start thinking about this very early on and not to just focus on the therapeutic molecule at hand, for example, or the mode of action, but really in how you want to manufacture your product. And also here you have different strategies that you could apply. So you want to be the fastest in the clinic because you want to be the first one to test that molecule in patients and demonstrate efficacy as early as possible? Or do you want to invest in a process which is a bit more sustainable and which can take you immediately to phase 2 and has less likely chances of, giving you product failures, manufacturing failures, or out of specification products? So these are clear also the decisions we have to make, and I think COVID has also impacted that field substantially. So we, of course, as a young company, we are not fully integrated and have our own capacities to manufacture. So we rely on external partners to manufacture the starting material for genetic modification, or to manufacture the TCR products for the patients. And what we've seen with COVID and, of course, the vaccine development is that slots for manufacturing fill up, even earlier than that was the case before. So the lead times to start your manufacturing have really become much more lengthier. And for us, that, of course, means that the time between discovery of a new receptor, a new molecule, a new therapeutic, to time to the clinic is much longer at the moment, and it's quite difficult to find slots for manufacturing for us at the moment. So that's one of the, in addition to the, of course, very obvious manufacturing hurdles, that's one that we're currently facing this year with the COVID situation.
Thanks, Elisa. You mentioned innovation, you mentioned BITES already, and Zach, maybe you can talk a little bit also about some other emerging areas of cancer biology, some novel targets, maybe even some innovation you have seen also at ESMO. Just to mention one example, antibody drug conjugates is now also not as an emerging field, but really also coming to the market. Would be great to hear your comments on that.
Sure, yeah, it's nice to see the continued innovation within the field. So you're right, ESMO, I think there were great data across the spate of modalities on the more classic front, small molecule targeted therapies. Amgen had their durability data associated with their KRAS G12C inhibitor in lung cancer, and it seems like the general consensus was that the durability was better than hoped for and would likely support a future registration. Within the IO space, I think we continue to see expansion of use of checkpoint inhibitors, Keytruda and Opdivo and others into either additional indications or earlier line settings or novel combinations with chemo. And there's now new IO targets being developed, things like anti-TIGIT. And then with respect to emerging modalities, yes, I think the ADCs had good. It's been a good couple of weeks for the ADC field, with Seattle Genetics entering into a large partnership with Merck, and then Immunomedics being acquired by Gilead. And I think both of those companies had data at ESMO that helped justify some of those deals. Other therapeutic categories that are becoming attractive are the protein degradation space, PROTAC. So some of those companies have recently gone public, and some have entered the clinic. So I think the field is looking forward to those. And then with respect to my own company's area of expertise, one of the things that I like to focus on when thinking about precision medicine in particular is kind of three questions. One is when we characterize a tumor, do we understand the driver? Do we understand what's causing these cells to grow? And the answer to that can be yes or no. If the answer is yes, is that driver druggable? So for instance, for a long time, the KRAS protein has not been considered druggable, but now companies like Mirati and Amgen and others have developed small molecules for certain, at least mutations or conformations of the KRAS protein, though not wild type. And then even for targets that are understood and are druggable, does resistance develop? And so EGFR is a classic example where we've had effective inhibitors, but eventually resistance developed at the T790M mutation location. Then companies like AZ developed a compound that was active against that, which was Tagrisso, which also had some nice data at ESMO, including in the adjuvant setting. So it's kind of this game we play in the cancer field, because the cancer cells, they're crafty and they continue to evolve around us. So first they have drivers, then the question is whether the drivers are druggable, and even when they are, do the cells find a way to resist those treatments? And again, that's where my company Boundless Bio comes in, because we're trying to understand the actual mechanisms by which the cells are evolving real time, and this ability to mutate their DNA when it does not reside on chromosomes, but instead resides on extra chromosomal DNA, that provides the cells a really powerful tool for essentially manipulating their genome, almost like a bacteria or a virus would. And so that requires a new therapeutic approach. We need to find a way to dismantle the cancer cell's ability to manipulate its genome in real time. And that's where we hope over the next couple of years to reveal to the cancer field some novel targets and novel ways of thinking about drugging tumors. So I'll pause there.
Mm-hmm. So Zach, would you say that this is really what is aiding the understanding, understanding of the molecular underpinnings of the tumor, even of a single cell? Is that what is helping to overcome some of the resistance we have seen previously, or some of the difficulties to drug certain targets that have so far been deemed as undruggable?
Yeah, I think the powerful tools that are now available to scientists here in the year 2020 versus what was available when I first started my career about 20 years ago. It is these tools and their precision that is enabling a new wave of treatments. So everything from deep genetic profiling, deep sequencing of patients, to broad population-wide efforts that help reveal rare mutations, rare drivers. You mentioned single cell sequencing. That's a very powerful tool that my company is employing now to understand heterogeneity or genomic variability within an actual tumor or a cohort of cells. There's also very precise genomic technologies or molecular technologies that allow you to vary from DNA sequencing to RNA sequencing to something called a TAC-seq that lets you visualize the chromatin accessibility. And when you start pulling together each of these tools and understanding, fluctuations in various molecules throughout the cell, you begin to identify correlations or patterns and kind of get a better understanding of what DNA sequence is driving what chromatin accessibility, which in turn it drives what RNA transcript levels, which finally results in what protein expression. When you can pull all that together, you get a much more clear picture as to perhaps where things are going haywire, or which seeming alterations actually matter for the physiology of the cell. And that does then reveal the insight into where you hypothesize that you want to try to therapeutically intervene. So I think it's a really exciting time in the field where we can bring all these tools to bear to inform very rational drug discovery.
That's fantastic. Zach and Dana, maybe you can comment a little bit on this fact that we, on the one hand, have now very, very precise patient and even cell-specific data. On the other hand, we have a lot of data. So how can artificial intelligence and machine learning be used to develop new therapies?
I'm happy to take a first crack at this, and I would love to hear Zach's thoughts as well. So I think there's a ton of promise with using machine learning approaches and artificial intelligence. One of the ways that we think a lot about it, to be completely transparent, I think we dipped our toes into the machine learning game later, and the main reason was the our focus was to build out reliable, I'll say, quote unquote, gold standard data. So data that is validated by humans, essentially. But with this sort of massive store of data that we've amassed, and this isn't Flatiron specific, I think across the data ecosystem now, we have so much data, to your point, that we're at a place where we can build reliable models, and we should be thinking about how to do so to kind of expand the reach of this data. So one use case that I think a lot about that's near and dear to my heart is in kind of this paradigm of precision oncology, where we're looking at increasingly rare populations, less than 1% of prevalence, right? It's hard to find these patients. And so one application is, can we use machine learning models to predict who might have a specific mutation or alteration of interest and better target our curation efforts? And so, right, for this use case of finding a needle in a haystack patient, right, we can use these models to look through, 2 million plus patients in our network, predict who might have the biomarker profile of interest, and then better target our abstraction resources in order to confirm whether the patient has that genomic profiling of interest. And so this is more of an efficiency play, I'll say, but it's a powerful one as we look to kind of curate clinical data and genomic data on these rare populations quickly. It allows us to do it a lot faster. Another area that we're seeing start to emerge, and I think this is still pretty exploratory, is, as we see new data types emerging. So in particular, digital pathology from reference labs, we're starting to see the kind of potential for leveraging machine learning models to comb through these new image types and better predict response or lack of response for particular therapies. So, while the first use case is more of an efficiency play and allows us to move more quickly to curate data, the second is, can we actually use these models to read data faster and actually develop algorithms and models to predict how a patient will respond? So I think there's a ton of promise there, and also a lot of uncharted territory that we're starting to enter into.
Yeah, there's definitely a lot of use cases. Also, we see it more and more even in very early stages of drug discovery, for instance, in the optimization of chemical structures. But I'm sure, Zach, you have some other emerging areas that you're seeing applications for AI and machine learning for.
Sure, I'll give a brief comment on that. I'm definitely not an expert in machine learning or AI, but I can say that our company has employed it once for a key component of what we do. So we are fortunate that a couple of our scientific founders and SAB members are prominent computational biologists who do have a lot of experience with machine learning. And the way that we use machine learning was to come up with a detection algorithm, a software that allows us to reliably detect when a tumor is driven by the ecDNA that I have been referring to throughout my remarks. And so I'll walk through the process by which they employed it. Originally, the observation of ecDNA, these are little circles of DNA that you can see inside the nucleus of the cell, and they are, as I mentioned earlier, physically distinct from a chromosome. So they're visualizable with what's called metaphase FISH, or you can see them with electron micrographs. So our founders found hundreds or thousands of cancer cell lines that were characterized by this observation of these ecDNA circles visually. And then they had hundreds of control tumor specimens that did not have the ecDNA. They deep sequenced all of these specimens using long-read optical mapping. So they then had these long DNA transcripts that represented either circular DNA or just normal chromosomal DNA. And then they used a machine learning algorithm and trained it with both the positive samples and the control samples to essentially understand, like, what are the characteristics of the DNA when it's circular as ecDNA versus when it's not? And can you reliably discern that? And then can you do that? So we then had the two sets, the positives and the negatives, based on long read sequencing. But we then reduced it to practice by then feeding the algorithm just normal transcripts from normal, like Illumina-based short read sequencing. And again, the algorithm was able to learn reliably when it was a circular form, and therefore it was an ecDNA, versus when it was normal chromosomal DNA. And so what we now have as a result of that machine learning process is a very reliable software package that we can take any tumor, any cancer specimen, just perform normal Illumina whole genome sequencing on that, and then using the raw transcript data and applying it to our algorithm, we can reliably determine whether that patient has an ecDNA or does not. And then that becomes then the criteria by which we determine our therapeutic methods. So hopefully that's a helpful real world case of where machine learning ultimately in this case helped come up with what will eventually be a companion diagnostic for therapeutic use.
Interesting. I'm sure we will see many, many more of these applications. Elisa, you mentioned that you were until relatively recently also in academia. Could you share with us also some of your insights on other emerging areas of cancer biology, novel targets, so innovation that you're seeing? And since you are in Europe, would be great to also get your European perspective here.
Sure. Yeah, so novel innovations that we're seeing, I think when we just spoke of targets, I think one of really the new wave of therapeutics after, again, with my cell therapy glasses on, it's now TCRs following the CAR T wave, basically, yeah. And I think the main reason for this is that people are looking to target tumor drivers, better antigens, which might allow to control tumor growth. And these are typically intracellular antigens. And the former CAR T formats, of course, have only allowed to target cell surface targets on the tumor cells and have been quite successful in doing that with the B-cell lineage markers. TCRs, as you know, on the contrary, they are able to also target any specific point mutation within a tumor cell, and by that we can address mutations like KRAS hotspot mutations, EGFR mutations. So everything that's basically considered a tumor driver and therefore a more valuable target, of course, is addressable to TCRs. And I think this is what we're seeing, like a huge wave in companies and also investments in the TCR space, specifically in solid tumors. Yeah, and we've had great financing rounds this summer, as you know, one in Tknife. The last one was announced last week for a Dutch company called Neogen, focusing on T-cell receptors on a patient individual mutation basis. But I think CARs are not falling behind, so people are trying to generate CARs now, which are seeing antigens in a very similar way, like a TCR, so on a HLA presented target. And of course, there's a chance to generate an antibody very specific for this HLA presented specific intracellular epitope. And so this is a recent development where more and more companies are looking to develop CAR formats that are not targeting cell surface antigens, but really intracellular antigens, including tumor driver mutations, which is, of course, interesting to see whether that pans out and really gives products which are specific enough and affine enough in the format of CARs. Yeah, I think this is really one of the key developments when look at the TCRs and cell therapy space.
Great to see that there's continuous innovation in Europe and in the field in general. Dana, do you want to share some of the emerging areas that you see in cancer biology, but also in the diagnostics field?
Yeah, I think the the piece we think a lot about is, right, what are the testing up modalities that are available to us for research based on what's used in real-world clinical care. One of the lenses that I think a lot about as well is not only the testing modalities, but also the evidence collection mechanism. So, you know, we are primarily focused on retrospective data collection. So, right, we are limited, as I mentioned earlier, by what's currently used in routine clinical care. But if we start to develop prospective collection mechanisms, we can get a bit more creative, right? If we can solve patient consent, we can, you know, we have a network of engaged sites with research capabilities. Can we build data sets prospectively to start to capture some of these more emerging modalities? So, you know, some of the directions that are exciting, right, is, you know, can we prospectively capture deeper sequencing like whole exome and whole transcriptome untargeted populations, right? We can only do that prospectively because we don't see as much kind of routine usage of that in the real world. Or we're starting to see some emerging assays in the early cancer space, right? We know that a lot of drug development is trending earlier. And so there are some interesting new technologies about monitoring in the early space and predicting, you know, when a cancer may become advanced or metastatic. And so can we start to move some of the, or see some of those technologies earlier in the cancer cycle by prospectively enrolling patients and using those technologies where appropriate. So I'm particularly excited about exploring more prospective collection mechanisms as a way to leverage and learn from some of these emerging diagnostic modalities.
That's great to see that innovation is going both earlier and also a little bit later. There's more and more combination therapies now that we're seeing also beyond chemo. Zach, what are you seeing in terms of more generally some emerging areas of cancer biology and novel targets, novel tools? You spoke a little bit about what you're seeing in your company.
Yeah, so I'll talk about maybe 4 categories of innovation in cancer biology and targets. So one we've talked a little bit about is drugging the historically undruggable targets. And so I think 2020 is really the year of KRAS. And so that's the shining light this year with multiple companies demonstrating proof of concept or entering the clinic either with direct KRAS inhibitors or even with combo therapies against that target and its pathway. There's been efforts against c-Myc over time, and recently there's a couple companies going after the p53 MDM2 protein-protein interaction, but these have all historically been undruggable, and now we're starting to see progress. So that definitely represents innovation. A second category I would discuss is, let's call it next generation or better chemistry for known targets and their resistance mutations. So I'd mentioned EGFR earlier and the T790 mutation, but other druggable protein targets like ALK and ROS and RET and BRAF. For each of these, the field has understood what are the common on-target resistance mutations from drug inhibition, and can we generate different compounds with different structures or that bind in a different way that overcome these resistance mutations. And I think we're seeing progress on each of those fronts. A third category then is protein degradation, which can actually address either of the first 2 categories, undruggable proteins or maybe overcoming resistance by just completely degrading the protein altogether. And I think over the next couple of years, we'll see how that field plays out. I know folks are optimistic. And then a final category I'll talk about is just new biology in general. And I think, you know, maybe one of the nice embodiments of this over the past few years has been the concepts of synthetic lethality, particularly in the DNA damage response arena. So that's been the advent of the PARP inhibitors, when against BRCA mutated tumors or tumors with homologous recombination deficiency. And I think some of those concepts are likely going to play out in the space where our company, Boundless, is playing, where we're seeing that the cells are relying upon extrachromosomal DNA to amplify certain oncogenes. And for us, instead of trying to therapeutically target the oncogene itself, we might be able to target some of the cellular machinery that is enabling the behavior of the extrachromosomal DNA. And if we take out that machinery, we may find that that indeed is synthetically lethal for these tumors that are relying on that machinery to ultimately propel their oncodriver. So I think there's been a ton of innovation within oncology across different targets, different modalities, and even just different concepts of thinking about how to drug the cell, whether you're trying to target something directly or more indirectly through a synthetically lethal approach. And it's been really nice to see each of those approaches enabled with proof of concept data and eventually drugs make it to market and helping patients. And so we hope to continue that march at Boundless.
Thank you, Zach. It's really encouraging to see that there's a lot of innovation both on the molecular level and also in some indications that are still showing a lot of unmet medical need. As we're concluding the webinar, Dana and Lisa, would you like to make one final remark each?
Should I start, Dana, or you,
yes.
you want to go first? Sure. Yeah, so from what I hear and what our discussion also is focused a lot in the recent months is we should move towards combinations. We need to evaluate combinations. We want to avoid patients relapsing from single targeting drugs and really what can we do also across fields in terms of, combining cell therapies with, for example, checkpoints or with specifically targeting drugs to achieve really the highest benefit for the patient and to avoid seeing patients relapsing. Yeah.
I agree with that. I think it's always going to be easier to take a drug that we know works and move it to new populations. And I think that and this kind of relates to a bit of the three-part framework that Zach was mentioning, right? But the part that I'm always going to care most about and be excited about is, how do we curate the data to support those strategies? And I think there's increasing, while there are significant limitations, I think there are increasing possibilities with real-world evidence to help us get there. And, where there are gaps, there will be kind of interesting ways of filling those gaps through alternative evidence generation, which I think the field we're seeing an inflection point where we're starting to move a lot faster there. So I think there's a lot of promise to be had.
Wonderful. Thank you very, very much to Elisa, Dana, and Zach for really, really insightful discussions spanning a very, very wide field, starting with diagnostics and the molecular underpinnings of oncology and cell therapy, even talking about some indications, and also spanning a wide geographic area from Berlin to San Francisco. Big thank you for a very interesting discussion, and I'm handing over to Sabrina for final remarks.
Yes, thank you, Stephanie and Dana and Elisa and Zach for this great session. Just a final word to everyone, and to let you know that there will be 2 more virtual partnering events before the end of the year, focusing on 2 critical therapeutic areas with great unmet need. So neuroscience will be next early November and infectious diseases early December. And registration is open at virtualpartnering.com. As we close this webinar, we would really appreciate your feedback. So if you could take 1 minute to answer the quick survey that will pop up on your screen, it would be fantastic. Again, we would like to thank you all for attending today and wish you a great event. with lots of new connections and collaborations in the oncology field. Thank you, everyone. Bye.